Imagine for a moment that you could shrink down to the size of a red blood cell and swim through the intricate network of blood vessels in your body. You'd find yourself navigating a maze of tiny, winding tubes, each one no wider than a human hair. This world, invisible to the naked eye, was first explored by a brilliant 17th-century scientist named Marcello Malpighi. His groundbreaking discoveries not only revealed the hidden complexity of the human body, but also opened up new frontiers in the study of life itself. Marcello Malpighi was born in 1628 in a small town near Bologna, Italy. He grew up in a time when the scientific world was on the cusp of a revolution. The old ways of thinking, based on the teachings of ancient philosophers like Galen and Aristotle, were being challenged by new methods of observation and experimentation. Malpighi, with his sharp mind and insatiable curiosity, was at the forefront of this revolution. One of Malpighi's most significant discoveries was the existence of capillaries, the tiny blood vessels that connect arteries and veins. Before Malpighi, scientists believed that blood simply flowed into open spaces in the body. But Malpighi, using a microscope, observed that blood was always contained within a network of tubules. This discovery was crucial because it helped explain how blood could efficiently distribute oxygen and nutrients to every cell in the body. To help others verify his findings, Malpighi provided detailed instructions on how to prepare and observe specimens. He described how to mount a frog's lung on a crystal plate, how to illuminate it, and how to use a microscope with 1 or 2 lenses to observe the intricate network of blood vessels. Even after the frog's heart had stopped beating, the flow of blood through these vessels could still be seen. This observation was a powerful demonstration of the continuous nature of the circulatory system. Malpighi's work on the frog's lung also shed light on the structure and function of the human lung. He noted that the network of blood vessels in the lung was similar to the network he observed in other animals, suggesting that the same principles of anatomy applied across different species. This idea of comparative anatomy was revolutionary, as it allowed scientists to draw analogies between the structures of different organisms and understand the underlying principles of life. But Malpighi's curiosity didn't stop at the frog's lung. He was driven to explore the microscopic world in all its forms. In the tongue, he discovered tiny taste buds, or papillae, and began to describe their function. He revealed the structure of glands, the distribution of gray matter in the brain, and the subtle structures of the cerebrum and cerebellum. He discovered the pigmentary layer of the skin, which gives our skin its color. His name is now attached to parts of the kidney and the spleen, which he was the first to describe in detail. Malpighi's work extended beyond the human body to the world of lower animals and insects, which were often overlooked by his contemporaries. He conducted a classic study of the silkworm, providing the first detailed treatise on the anatomy of an invertebrate. The silkworm's intricate system of tracheae, or air tubes, helped him understand the process of respiration in these tiny creatures. He even compared the tracheal system of insects to the cell structure of plants, laying the foundation for the field of plant anatomy. Despite his bias against theoretical speculation, Malpighi was not afraid to draw grand conclusions from his observations. He observed that the more perfect an organism, the smaller its respiratory organs. Plants, which have their respiratory organs spread all over their surface, and insects, which have tracheae throughout their bodies, have extensive respiratory systems. In contrast, higher animals like humans have relatively small lungs, suggesting that the efficiency of these organs increases with the complexity of the organism. Malpighi's work was not without its challenges. He faced criticism and envy from his medical colleagues, who were often resistant to new ideas. Some even accused him of using the microscope to distort reality. But Malpighi's dedication to empirical observation and his willingness to follow the evidence wherever it led him ultimately won him the respect of the scientific community. One of the most significant moments in Malpighi's career came in 1689, when a formal indictment was pronounced against him at the library of the Friars Servants of the Blessed Mary in Rome. Fourth Assise, defended by one of his own students, condemned his work as useless. They argued that the anatomy of small, internal structures was of no use to physicians, that the idea of capillaries was untrue, and that the study of insects and plants was of no practical value. Malpighi High's response was to continue his work, knowing that the truth would eventually be recognized. Malpighi's life was marked by both triumphs and tragedies. His long and fruitful collaboration with his friend and colleague Giovanni Borelli was strained by a dispute over the discovery of the spiral structure in the fibers of a macerated beef heart. The two men, once close friends, became locked in an acrimonious debate over who had made the discovery first. This dispute was a painful reminder of the competitive nature of scientific discovery, even among those who had once been the closest of colleagues. When Malpighi died in 1694, he left behind a legacy that would change the course of science. His colleagues at the Royal Society in London recognized his contributions with a detailed obituary, noting the deformity of his right kidney, the enlarged heart, and the broken blood vessel that caused his fatal stroke. They praised him for his dedication to serious study and for his role in advancing the commonwealth of learning. Malpighi's work was part of a broader movement in the 17th century that transformed the way science was conducted. The idea of a parliament of scientists began to take shape, with new forms for the exchange of ideas and the sharing of discoveries. One of the key figures in this movement was Marin Mersenne, a French monk and scientist who lived near the Place des Vosges in Paris. Mersenne's personal charm and generosity made his monastery a center of scientific life, attracting some of the most brilliant minds of the day, including Galileo, Descartes, and Pascal. Mersenne's network of correspondence extended across Europe, from London to Constantinople, bringing together the latest scientific discoveries and theories. He was known for his ability to mediate disputes and bring scientists together, despite their often irascible and competitive natures. Mersenne's work laid the foundation for the formation of formal scientific societies, like the Royal Society in London, which would become the model for scientific collaboration in the centuries to come. The Royal Society, founded in 1660, was a product of this new spirit of scientific inquiry. One of its key figures was Henry Oldenburg, a German-born diplomat and scientist who had studied in Bremen and Utrecht. Oldenburg's fluency in multiple languages and his personal charm made him an ideal organizer for the new society. He had already established a network of scientific contacts in England, including the chemist Robert Boyle and the philosopher John Milton. Oldenburg's role was to facilitate communication and collaboration among scientists, ensuring that new discoveries were shared and discussed. The Royal Society's motto, Nullius in verba, take nobody's word for it, reflected the new emphasis on empirical observation and experimental evidence. The Society's members, including Boyle, Isaac Newton, and Christopher Wren, met regularly to discuss their work and share their findings. The Society also published the first scientific journal, the Philosophical Transactions, which became a model for scientific communication. The formation of the Royal Society and other scientific societies marked a significant shift in the way science was conducted. No longer was science the domain of isolated geniuses working in their private laboratories. Instead, it became a collaborative effort, with scientists from different countries and backgrounds coming together to share their discoveries and build on each other's work. This new model of scientific collaboration would accelerate the pace of discovery and lay the foundation for the scientific revolution. Marcello Malpighi's legacy extends far beyond his own discoveries. His work on the microscopic world and his contributions to comparative anatomy opened new avenues of research and inspired generations of scientists. The spirit of curiosity and empirical observation that he embodied continues to drive scientific inquiry today. As we continue to explore the hidden worlds within our bodies and the natural world around us, we are reminded of the enduring impact of Malpighi's groundbreaking work. In the end, Malpighi's life and work remind us that the pursuit of knowledge is a journey of endless discovery. The microscopic worlds he revealed, the intricate networks of blood vessels, and the hidden structures of the human body and other organisms are a testament to the beauty and complexity of life. Malpighi's legacy is not just in the discoveries he made, but in the way he inspired others to look deeper, to question, and to explore. His work continues to inspire scientists and students today, reminding us that the most profound truths often lie in the smallest details,